Hi there, Mindsetters. My name is Megan, and welcome grade 11s to today's Life Sciences show. Today, we have a special revision lesson prepared especially for you. We'll be selecting highlights from the lessons shown early in the term to help you revise for your end-of-term tests. You can download the notes for today's show on learnextra.co.za/live.
I'm here. Now, I brought a couple of things with me, right? So it was just to actually see what is going on, right? And it's always nice. Now, you got to understand that I had to get these things going. Not all of them, but one thing is getting to me is that they don't smell very nice. Right. So what I'm going to do is, if you come closer, if you don't mind, so please come a bit closer, and you can understand.
He's zooming in.
Is he? Yeah, no, but I need him here.
That's where he is.
You kidding me?
That's where he is. Trust me.
Okay. Now, if we have a look at it, there is different little things here. Now, what I've got here is I've got a couple of things for fungi, right? Now, that is the next piece that we're looking at, right? So we've got fungi, because fungi is quite easy to get, right? So that is something you've got to remember. Now, this one was left over from, what's that stuff? Mousse. Now, that is so cool, but doesn't smell very nice when you open. So all these things that you guys pull out of your fridges and put into the dustbin, don't think that when it's in the fridge, you are perfectly safe. You must understand that bacteria, viruses, fungi, all of those, they can handle certain type of temperatures. Some can handle low temperatures, some can handle very, very high temperatures. So don't make the mistake of, if it's in the fridge, it's safe. So please remember that. Now, let's have a look at this. If I have a look at these things, right, I've got a nice white one here. Now, this looks very nice. It looks like Father Christmas to me. To me, that's what it looks like a beard, a nice snow white beard, right? This one. So the next one, if you can see, it's nice and green. It's the type of fungi that most probably can photosynthesize, right? So we've got the white one. We've got another white one. I don't want to touch it too much. So I'm going to open it up. You can see it actually sticks. If I poke it, they stick together. Can you understand what I'm saying? Right? Sticks, and they form one thing. And then right here, if you can have a look at it, this piece over here, the specific piece over here, I want you to have a good look at it, right? Now, that part there is a very nice one, because that there shows me different parts of fungi that you need to know, right? It's got a sporangiopore, it's got sporangium, right? And those are one of the nicest things. When I saw that, I thought, whoa, that's going to be perfect for the lesson, right? So that is quite cool. The next thing I have here, which is, you have no, you had to smell this. This smells like penicillin. Okay? This is penicillin at its best. So if you have that, sorry, my nose is itching. If you have that, right, and you are allergic to penicillin, this you must stay away from. This is your bread mold, right? This is bread mold to the max. I mean, I kept this for a long time just to show you, right? So this is bread mold. I got moaned at my wife, so I had to keep this outside, right? So if you have a look at it, it's nice, it's green. If I could get any closer, I don't want to take it out of the bag because it can be dangerous. I don't know if Kat is
No, I'm okay. I don't want to
so she's
touch
happy with it, right? So I've made it nicely, and it's there perfectly for you guys. Hopefully you can see, and it's very poisonous to people that need insulin, because this has got it, right? And then lastly, if you can have a look, I brought a mushroom. Now, these are quite cool. These are things that you can actually eat, right? And this is a fungus. It's a fungus, and you can eat it. So what I did was I cut it in half. There we go, because you need to know what the inside looks like. And you're going to learn things about, what was those words? Hyphae, right? And rhizoids. And you're going to know about your cytoles. What's it? It's your cytoles, which are specific things. Now, this hyphae, right? Hyphaes are specific parts. And I'm going to go show you down on the board exactly what they are. Now, please remember, fungi you can eat. So don't stress too much about certain things. But something about...
You might want to cook that first. I don't know.
No, I'm not worried
Are
about
you
things.
okay?
Now, understand something about fungi, right? Especially this one, okay? Do not, I repeat, do not just go and eat these mushrooms, any mushrooms. Well, these mushrooms are fine because I bought them from fruit and veg, right? But if you're walking along, stay away from mushrooms. They are highly, they can kill you. I don't know how else to say it. highly, they can kill you, right? So they will kill you, or they make your life hell, or they make you feel so nauseous and so sick, they can be so bad for you. So please, if you are not sure about a mushroom, if you're not 100% sure that you can eat that mushroom, don't dare. Not even the professionals out in the open will eat mushrooms without them knowing.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision. Over to you.
This came out of an exemplar. It's a nice question, and it's something that you can put your head around and start thinking. Okay, so what they did, the question, I'm not going to read it word for word, but you can read it. But what I'm going to say is that there was somebody with a throat infection, right? So he went to the doctor, and they took a swab of this infection, this bacteria, and they put it on agar. Now, please don't forget, agar, where it is, there it is. Agar is very cool because you can grow bacteria on it, right? So they put on agar in a petri dish. That's another big word, right? And they let it grow, right? But they want to see what bacteria it is and what antibiotic can kill it. That's what they did. So what they did is they put it on a petri dish. There it is. And that looks like German at the moment. So what I'm going to do is I'm going to bring it down so you can have a look at the key. Okay, now, there's the key. It's a little bit small. Okay, so all the black, the simple black one, is the zone with no bacteria growth. Okay, and all the other funny shaded one is where bacteria grew. So this is bacteria, right? This is no bacteria. Right, are you all with me? So this is where the bacteria hasn't grown. You all happy with that? So hopefully I'm going to switch to and from the slides all the time so that you can get it. So let's have a look at it. Okay, state one difference in the active between antibiotic B and F. So explain what's the difference between B and F. So let me go back. What's the difference between B and F? Let's have a look. B and F. There's B. Look at the size of this. There's B and there's F. Okay, sorry, a little bit bigger. Now, what does that tell you? If B has got this whole black section, and remember, black means there is no bacteria growth. If B is like this size and F is like this size, what does it tell you? Automatically, it should tell you that the antibiotic, all these little strands, these pieces here, where all the letters are, they tried different types of antibiotics. I forgot to tell you that, so sorry. Right, so they've tried different types of antibiotics. We're all with me? Okay, so they put this antibiotic there, this one there, that one there. So at F, there's a specific antibiotic. On B, there's a specific antibiotic. So what does that tell you? What does that tell you about the antibiotic? Right, to me, that tells me that antibiotic B is very effective to this virus, this bacteria. It kills this bacteria. Remember, the bacteria started all in there. They put the antibiotics there, and it killed everything that way. Or they put the antibiotics there, and it didn't grow towards it, so it stops it from reproducing. Right. This one, they could come a little bit closer. So what does that tell you? Okay, it means it does stop it, but not as well as B. So the question was, let me get back there, state one difference in activity between antibiotics B and F. B is a much better antibiotic than F to this type of bacteria. Cool. Now, let's have a look at the next one. The patient was known to be allergic to B. Oh, now we got a problem, right? So the best antibiotics that we've got, this patient decides to be the only one to be allergic to this antibiotic. So now the doctor cannot give this antibiotics to this person, right? So what the doctor goes up and says, well, now we're in trouble, so cross this one out. We're not going to use it. There's nothing we can do. Pick another one. Which one would you pick? Would you pick A, which is very small, F, which we spoke about earlier, or would we have a look at C, D, or, E, D, or C? Take a guess. Me, personally, I would definitely go with E, because have a look. It kills the bacteria, or the bacteria can't get close to it, not as well as B, but enough, if you get what I'm trying to say. It's the next best antibiotic. So let's go have a look at the question again. Right, there we go. The patient was known to be allergic to antibiotic B. Let's see. Which antibiotic should the patient be given? Come on. Not even before I looked at the question, I could find out what the answers. Can you remember what it was? It was definitely, one more time, it was definitely E. Definitely E. It's the best one there after B. Okay? Then, let's see. The next one was, explain your answer to your question. You know what? I think you guys are getting this quite easily. I think. Okay? Why would you choose E? Because it is the one that had less bacteria closer to the antibiotics. It's the one that was the biggest area of no bacteria growth after B. B is the best, E was the second best. I'm sure you could understand that from the picture. Right. You're all with me so far. I think this is a stunning question, and I promise you, this type of question is very good for you. You need to have a look for these questions, because they like asking this, right? It's like a hands-on type of thing, which is nice, right? Let's have a look at the next question. If I can get there. There we go. Here we go. The organism causing the infection seems to be resistant to 2 of these antibiotics. Resistant. Now remember, you can't just take any antibiotic. So you can't just go home, you're feeling sick. Oh, I remember I took those antibiotics, and let's take those again. First of all, you didn't finish your antibiotics, which is very bad. Okay? You need to finish all antibiotics. The strain completely so we can kill that thing in your body. The second of all, how do you know that bacteria will be able to be killed by that antibiotic? Okay? So you need to make sure you can't just go and take any antibiotic. It's very bad for you. Okay, so let's go have a look. Now I know that it's, if I have a look at it, there it is. Which ones, out of all of them, A to D, or to F, A to F, which ones are immune, which ones of the antibiotics did not work on the bacteria? Let's have a look. I can't erase it, so just have a look. F is nice and big. Oh wait, that didn't work. F is nice, and I'll put them back. There's F, right? There's E, okay? There's D, D you can still see. There's C, and there's A. Okay, have a look at that. Can you see that? Which ones would they be? I think, if you know your stuff carefully, remember, this black spot over here, this specific black spot over there, okay, that's just where the antibiotic was put down onto the agar, right? So that means this antibiotics is not growing over there. Do you understand what I'm trying to say? So it's definitely, if I have a good look, it's definitely, I just want to get rid of that, it's definitely A and C. A and C are the 2 where the bacteria is grown all the way to the end. All the way to the end. So that is a nice, easy answer. So if I go back and I have a look at that question again, okay, which 2 antibiotics are referred to as the statement, the organism causes infection seems to be resistant to 2 of these antibiotics. Can you remember which ones they are? They're A and C. Remember? They couldn't get close enough. And then it says, which 2 antibiotics are referred to in the statement above? Let's see. Let's have a good. Organism causes infection seems to be resistant to 2 of these antibiotics. Which 2 antibiotics are referred to in the statement above? It is A and C. So that is the question, not that. That was just a statement. Okay? The next one, explain your answer to question 1. Have you noticed how you get an answer and they always ask you to explain it? Okay? The reason for that is it's not just knowing what is going on. You need to understand it. Understanding is very important in biology. So make sure you listen to your teachers. Because if you don't listen to your teachers, you can read out of a textbook. You need to understand it, and that's what your teacher does. She helps you understand it. Now, this guy says that these two antibiotics are resistant, or the bacteria is resistant to these specific antibiotics. Which ones are they? They were A and C. Why is it? Because the bacteria grows all the way to the outside of the area, or all the way to the antibiotic itself. The antibiotic did not stop any of the growth. Okay, so hopefully you're understanding that. Okay, I'm happy with that because I know that you guys are listening and you're getting there. Right, so is there any more? That's it. The patient was given a 5-day course of appropriate antibiotic. Remember, they couldn't take B. He had to take something else. Can you remember? So it's the appropriate antibiotic. Explain why it is important to finish. Remember, I moaned at you earlier. Finish the course of antibiotics when you are feeling better. Now, understand this. When we take antibiotics, I take it now and I'm killing off the bacteria. Right. Now, there's very little in me. Let's say there's 10 instead of like 5,000, right? So there's 10 left. We've killed off all the others, right? And those 5,000 are not doing anything to you yet. So you think, I'm nice and strong, and we're cool. We can go jogging again. We can do everything. Ma, keep the antibiotics for the next time we need it. We're going to be cool. That's a bit of a problem, right? Because what happens is that bacteria is still inside you. So they're either going to flare up again, which means they're going to infect you all over again, or they're going to change their DNA. So they get used to this antibiotic. And because they get used to this antibiotic, they can change, which means this antibiotic is not going to work. And especially if it changes and then gets given to another person, what's going to happen? We're going to start a new bacteria, and that's going to grow, and we're going to have to get new antibiotics. They're going to have to do more work in the labs. So do you understand where I'm coming from? Very, very, very important to finish your antibiotics. You need to kill all the bacteria before you stop your antibiotics. That's why he gives you 5 days and not 3. If it was going to last 3 days, he'd give you 3 days. Finish it. No matter how well you're feeling, finish the antibiotics. We cool with this? Trust me, my mom used to moan at me often because of the exact same thing.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision.
So let's have a look at this. I'm going to put a diagram up because what I've done is I've put some questions on and I've been nice. Okay, I'm going to really be nice. I've made them difficult. I think that's fair, hey?
So you've been terrible.
No, nice. The more difficult the question, the better it is for you because you learn more. So what I've done is I'm going to give you this one. So it just says the diagram below represents a spore in a radiophyte. Now have a look. Have a good look at that. Now, do you know what that is? Because if I had a look at that in the beginning, I would think, oh my word, that is not going to work for me. So let me just put it out there. I want you to have a good look at this. So if I had to color this in green, right? And this part also in green, right? And this piece will be blackish, according to what I saw. This is the sori. Can you remember the sori? The sori is where we kept all the, can you remember, spores? Okay, now let's have a look. Questions. Questions are always good. Name the plant in which the sori is found. The source is found. Oh, please, come on. Guys, look at this. Before we do anything, we actually need some color. It's here somewhere. There it is. But you might not be able to see it on the board, but there it is. Okay? It is the fern. Can you remember the fern? The fern is very important. This happens in the fern. That was that old leaf that I showed you with those black dots on the back. That's what that is. Okay, cool. So the source is a fern. That's where it comes from. Okay? Don't forget it. Have a good look at this. They like sending this to you. Okay, cool. Next, if I bring it down, identify parts numbered 1 and 6. 1 and 6. Now, in actual fact, there's a piece missing on this. Now, what I'm going to do is I'm actually going to try and draw it for you. Not very big, but when I got the picture, I'm sure there was something missing. So what happens is there's something over here, and it just going to go like this. I'm just going to color it in, okay, just so you can see. Now, that there is normally like a vein. I can only say it, vein. It's the best way of putting it, okay? That there, okay, is your vascular bundles, your tissues. That's the vascular tissue, the xylem and the phloem. Those are very important because if they're not there, right, these things are not going to get food. They're not going to get water. Do you understand? So it's very important to get those. Next thing, they need to give food to all these little things here on the side, which we're going to get to in a few minutes. Okay, now 6. Now 6 over here, hopefully I can get it to draw, is that piece there. Okay, it's this little black dot in the middle there. Now, if there's a little black dot and there's something holding a little black dot, we need to understand which is which, right? The thing that's holding the little black dot, okay, is like a sporangium. It protects the black dot. The black dot is a spore. Okay, the spore is what gets let out, and then we make the gametophyte generation. Hey, how cool is that? This is a spore, comes from a sporangium. Okay, so very, very important. Okay, so we've organized that. The first one was vascular bundle. Next one was the spores. Very simple, simple answers. Okay. Number C. What is the function of part numbered 3 and 6? 3, 3, 3, 3, 3. Okay, what is the function of this thing? Me and this pen today, we're loving each other. What is the function of this piece? Okay, what is the function of the sporangium? Okay, the sporangium is the protector. It looks after the spores. It keeps the spores in one place so that it can grow up and mature. I mean, it's like a school, right? Don't put this on your paper. I'm just trying to make you remember it. This here protects those spores until they're old enough to go into the big wide world and do what they need to do. Right. That's the best way to put it. What do you think, Indiana? I think that's perfect. They protect the spores until they are good enough to be blasted off and out there so that they can make a way and make a new plant, right? So that's the job of 3. And the other one, if I'm not mistaken, was 6, but I don't want to just draw, it is 6. What is the job of 6? What is the job of the spores? It's for reproduction. We need to make new plants. The more plants we make, the better it is for that species to survive. Please remember that. It's very important. Now, the spore's job is to actually to survive where we're going. Right. So, you're all with me there. That's quite nice. And it was very, very, very simple. Next one. I'm sure there was more. No, there wasn't any more. We're going to go to the next one. There was. Trying to think if I went there. That's it. Is this the sporophyte or gametophyte generation? Provide a reason for your answer. Now, you haven't learned it yet. I will tell you next week. But when we have a look at this thing, I'm going to draw it on this side. So deal with me. This is, of course, the fern. And if we have the fern going, the fern has 2 different generations. Can you remember? The gametophyte generation and the sporophyte generation. It just told you here. Don't forget these 2 words, right? Now, those 2 things are very important. Gametes means, can you remember gametes? If I look at me, it's the sperm and the egg. If you look at flowering plants, it is the pollen and the egg. You're all with me. So that's the gametophyte generation. And the sporophyte generation is the adult plant. Right, so if I had to go and draw something, you've got to remember this very carefully, that this plant, what's so nice about it is the gametophyte generation is completely different to the sporophyte generation. Now, the gametophyte generation, I think the girls like this one. I'm hoping the girls like this one because this is the plant that looks like this. This is a piece that comes out of it. It gives it one of these. You won't believe it. Come on, draw. What does that look like?
What's a heart?
It's a heart.
Aw.
Love. Can you remember love? You said share the love.
Share the love, guys.
That there is a prothallus. Now, a prothallus is, can you believe it, is the gametophyte generation of the fern. Right. So this guy here actually gets, now, please understand where I'm coming from. Roots, it's not really roots, but roots coming out the bottom, and it gets little plant going up there, which is the gametophyte, the gametophyte, and that there meets up with another gametophyte, and then they make babies, and it's this big plant. How cool is that? So the question was, is this a sporophyte or a gametophyte generation? The sporophyte generation has got to make spores to make the gametophyte generation. How cool is that? Sporophyte generation makes spores to make the gametophyte generation. So, what are these things? Can you remember? They are spores. Okay? So, if there is spores present here, what makes the spores? The sporophyte generation needs to make the spores to make the gametophyte generation. So, the question is this a sporophyte or a gametophyte? It is sporophyte generation. Explain your answers. Because it has spores in the leaf. It has the sori. Remember, this is the sori. This is the part of the plant underneath the leaf with all those dots to let the spores go. Cool. So there's a nice question, and I'm sure your teacher's going to ask you that one day. What does the term haploid mean? You see, I told you must listen. Haploid. I'm sure you wrote that down. Haploid, can you remember? Half the amount of the original chromosomes. So, in your body, right? You have cells for Africa and America and New Zealand and all of them, right? That's how many cells we've got. Cool? And they are all what type of cells? They are all diploid. Remember, diploid meaning how many? It's 2n is diploid. They want to know what haploid is, right, which is n. Okay? Diploid means 2 n's. Okay? It means if n is 6, this will be 2 6's, which is 12. There we go. So you got it. So n is half the original chromosomes. This is the sporophyte generation. This is the gametophyte generation. That means haploid is half the amount of the original chromosome. We all got it. You cannot forget that. It's very, very important. And I know the next question is about where can we find it on the plant. Let's see. Let's see. Write down the number of the haploid structure. You know, and you must understand, I love things like this. Have a look. You tell me, where do I find the haploid structure there? Haploid, remember, this is all made up of cells. Remember, cells are everywhere. This is made up of cells. That is made up of cells. All of this is made up of cells. Show me or give me the number of the haploid structure in that thing. Because there is a haploid structure. Can you tell me which it is? This, to me, is these little black things over here. You know the little black thing over here? The spores, right? Now, spores are haploid. Cool. They're the ones that go make the gametophyte generation. The gametophyte generation is haploid. How do we make it? Through the through the spores. Right. You're all getting this. I hope it's making sense. I hope it's clicking. It's the best thing in the world, right? Don't ever tell your teacher that plants are boring. They're not. They're as interesting as you make them. It's always as interesting as you make them. When you learn about a plant, go out, go find it, go and take a piece off, have a good look at it, cut it, see what it looks like, try find a microscope, try find something and have a good look at it. Because the more you see, the more you learn, the more you understand. Okay? Anybody can understand something, well, anybody can learn something off by heart. You're going to forget it. You must understand it to be able to answer it at the end of the year. Okay? Understanding is the key thing compared to anything. Anybody can pass a test. Anybody can pass a test now. But in 6 weeks' time, 7 weeks' time, when you can't remember it and you got to go study it, you go, oh my word, I'm going to fail because you didn't understand it. How do you understand it? Find the love in it. Right? The more you love it, the better it becomes, the more you understand it. If you understand it, good marks. That's what I tell my boys. Right, now, can you tell me what that structure is? Okay? It's the diagram shows part of a branch of a... I did that for a reason. Can you believe it? That's just... Remember, you've learned classifications, right? And you've got you're going to learn about classifications like Chordata and Monera and all of those kingdoms and the plants and all. So we'll get there. But I did that just to scare you a bit. Okay? Now, what is this little thing over here? I've actually made sure that I could try and confuse you. So let's look at the question. Okay. Identify parts labeled 1 and 5. 1. Oh, whoa. You see, I can't even read. 4 and 5. There's 4 and there's 5. Okay? They're on both things. So what I did was, we took this piece over here, okay, and I like cut it and I stuck it over here so that I can make you see exactly what is going on. Now, what is this? How are you going to notice it? Look here. Look at the structure here. Can you see the structure? What is that structure called? It is a cone. So what is this? This is a gymnosperm. Okay, don't forget it. Gymnosperm because we got a cone. Cool. So 4 and 5, there it is. This is what happens if you look deep inside. You've got this little thing here, and if you look at this carefully, this piece that goes up here are actually the leaves. They're the needles that come out. But before they come out, they're on this little stalk over here. And that little stalk over there is called a dwarf stalk. Okay? It's a dwarf stalk. It's small. It's very small, so it's a dwarf and it's a stalk. The second thing, number 5, is they're pointing at these. You see that on that, you see little arrows like that, right? Those there are scale leaves. Okay, remember, scale leaves are just there for protection and to help it and keep everything together and so nothing hurts it. That's the scale leaves. Okay, so 4 is dwarf stalk and 5 is your scale leaves. Right, now, let's go on to the next question. Write down the number of the part in which, this is cool. This is cool. I like this question. This is, microspores are produced. Microspores. Now, they didn't say eggs, right? They said microspores. So if I bring this all the way up again, look at it. This is the female part, right? Can you remember I mentioned the male part? There it is. There's the male part of the plant. Now, that means that they make microspores. In other words, the male gamete. If they make the male gamete, right, they make microspores. Micro meaning very small, and spores meaning the part that comes from a sporangium, right? That's where you're going to get the sperm or the egg. I like using sperm because we identify to it, right? So the pollen, same way of looking at it, right? So you've got that one. Microspore, so which one would it be? Would it be 6? No, it will be 1. It will be the male part, right? So let's have a look at the next one. Seeds are produced in the, come on, it's those big things that fall on the floor, and sometimes we put them in fires because they burn quite nicely, right? It is the cones themselves. They make the seeds. Okay. Lastly, photosynthesis, it will be in the pins or the needles. You know those needles? The needles, they have chloroplasts. The next one, unlimited growth can be taken in. Now, you learnt about plants, right? And remember, where does plants grow from? It grows from the tips or from the growing points. On this piece here, right, you get your meristematic tissue or your apical meristem or your apical bud makes it grow that way. Cool. So that is where all the growth is going to come from. Okay, we all happy? Let's have a look at the next one. What is the average lifespan of part labeled 4? What is the average lifespan of number 4? That's just this piece here, lifespan. You know, every year we go and it comes to during summer and then it goes to winter, and what happens to the leaves? Indiana, can you tell me what happens to the leaves?
They fall off.
They fall off. That's why it's called fall, right? So they fall off. So how long do you think these things last for? How long do you think, Indiana?
A season,
Okay,
3 months.
that is it. 3 months, a season. Wrong.
Wrong.
I knew I'd catch you for once in my life.
It was a good guess.
It was a
right?
good guess.
Yeah.
But, okay, but this is 3 years.
Let's go to a short break now. Remember, if you are struggling and you need help, remember to post your comments and questions on our Facebook page, facebook.com/LearnExtra, or on Twitter @LearnExtra. Don't go away. We'll be right back. Welcome back, guys. Let's get into some more revision. Over to you.
I had got some questions from Solutions for All, Life Science, right? And they had some nice questions because these are the type of questions you're going to be asked. Okay, now the first question, as you can read on the board, right? I'm going to highlight certain things. In what way does sperm in moss and ferns, okay, differ from those in spermatophytes? Now, those are big words, huge words. Okay, so automatically they've told you we're looking at a moss and we're looking at ferns. Cool, so we've got those two. But what is a spermatophyte? Okay, spermatophyte means they're not going to go through alternation of generation. They're not going to make spores. Can you remember I said the capsule or whatever spits out all the spores and the spores grow into like the prothallus and that, right? These have only got sperm. Okay, so now, first of all, what is the difference? When you get to moss, right, and ferns, you need water. So you will notice that moss and ferns always grow where there's a lot of water. If you have a look where you find moss, it's normally where things are leaking water so that they're over water. Water's over them, right? And if that happens, then the sperm can get to the female's part by swimming. They've got flagella, which means movable parts that actually move and make them swim. So if I'm not mistaken, a moss's one looks something like that, right? And a fern's one looks, you won't believe it, it looks funny little curls, right? And then at the back here, it like makes a huge squiggle to swim with, if my memory isn't serving me right. But look at the big squiggles. Those are the main things. It actually makes it swim, right? Now, spermatophytes, these brothers, right, they've got stuff called pollen. Right, now what happens is we get this big pollen grain, and inside here we've got the sperm cells, okay, the two little sperm cells. So this thing covers them. That's what those, you know when you look at big flowers and you get that yellow stuff. Hey, Indy, can you remember what pollen is?
Pollen, of
That's
course. It's the stuff that makes me sneeze and I'm highly allergic.
Nice.
Ah.
And bees love the stuff because they don't actually like it. They want the nectar, but it goes all over them and they go from one plant to another plant and they can do it that way. So that is the big difference. These swim in water, these don't. Okay, that's the biggest difference. Now, if I have a look at the next one, what is the reason, okay, what is the reason for the difference in structure to the sperm mentioned in the question above? Now, I make sure I answer my questions perfectly. And if you've noticed, I've already answered this question in the top. I said to you, the reason why it is like this is because the moss and the fern, actually the sperm has to swim in water. Now think about it. If there is no water, can the moss and the fern reproduce sexually? Indy, do you think you can tell me? If there's no water,
No.
do you think that the moss or the fern can reproduce sexually? What do you think? Yes or no? Just a simple answer. Take a guess.
No.
Perfect. That's a lovely answer. No, because the sperm cannot swim to the egg. Right. It cannot get there. It needs the water. That's the first thing. Second thing, if you have a look at the spermatophyte, they have pollen. So pollen can flow in the wind, right? It needs to be light and fluffy. It can go into the wind. It's got to be carried by animals. It's like bees and that. So you must understand it's completely different. Okay, so the structure is completely different because sperm of the moss and the fern have to swim in water where the spermatophytes don't. Okay, that's a nice question. I like that question. The next one, as soon as I get there, why do watch this, this is cool, why do gymnosperms and grasses produce more pollen, right, than spermatophytes? Okay, now, why do plants like this produce a lot of pollen? So this is the reason why Indy sneezes all the time when it gets to pollination time, right? This is the main reason. So you want to get rid of it, get rid of all the grass and everything like that, and she'll feel much better. Okay, this is very important. Now, if I'm going to do this. I have this big male cone. Okay, I'm just going to make it a big male cone over here, right? And we're talking about the gymnosperms, right? And over here on this tree, right, there we go. We have another branch, and here is the female cone. Okay, the female cone. We're all happy with that. You get that? Okay, now, if I take this thing, it's just got full of pollen and it goes mad. That's so cool, right? And it has a lot of pollen. Now, it needs to get all this pollen, all that pollen, into this female part. Now, remember, let's have a look at a cone of the female, right? It does this. Can you all see what I'm trying to do? It does this. And if I come back, and it does that again. And there we go. That's what a female thing looks like. And we need to get the sperm all the way into there. Hey? That is going to be hell. So what he needs to do is, he doesn't just go, here, take your share. There's a couple for you. Hey? And he has like 3 for you, just in case. There's 2 spare.
Oh,
right? It's not like that. What it she does is, I mean, think about it. If you want to wet Indy, if I wanted to take Indy and wet her, okay, and I want to make sure she's wet, how many drops am I going to throw on her? 2 or 3? That's like catch. What happens if I miss? That doesn't help, right? I mean, I need to take a bucket of water. You get what I'm trying to say? If I take a bucket of water, how much am I going to miss? If I take a teaspoon of water and throw it at her, she's going to go, missed, right? If I take a bucket, she's going to go, and she's going to be sopping. You get my point? So the more there is, the more chance it has of getting inside, okay? That is the biggest reason it uses wind pollination. If we use wind pollination, it lets the wind take the pollen and take it to the female. But the wind doesn't know where the female is. It doesn't know the female, right? It takes it and it moves it all the way to the female part. But now only 4 sperm get there instead of 10. You get what I'm trying to say? We need to make thousands of sperm to make sure that at least 10 get there. Okay, do you understand? That's why they make so much pollen. That is the exact reason why, if we have a look at it, you have your grasses and your sperm, your grasses and your gametophytes, your gymnosperms actually have so much pollen. Okay, I think that's a stunning question. It's a very nice question. A lot of people are going to ask that question. Now, I'm going to go on to the next one. Okay, I want to know. Let me get my pen out. I'm getting this hang of green, right? How have the spermatophytes achieved the independence from water for reproduction? Now, remember I said to you that your moss and your fern need water. You understand this? So what do? We make it a more complex animal, right? We want this tree to grow. We want to actually make a new plant, right? So to be able to do that, you normally need water if it's moss and ferns, right? So now we decide, it's not going to work for us. We need, no matter what, we need there to be sperm and an egg to create a new plant, okay? Whether there's water or not. So what actually happens is we put it in a pollen grain. Okay, we've taken this locomotion of water away, and we've just put both of them in the pollen grain. If it's in the pollen grain, right, then it just gets taken to the female part, and all it has to do is dig a tube all the way down to the female. That's all it does. It doesn't have to swim. It doesn't have to go find anything. It gets transported in a car. I mean, think about it. The pollen is a car for these little things, and it goes all the way to the female part, lands on top there, and it digs a hole down. That's easy. That is so cool. It makes sure that it happens. You get what I mean? So it doesn't need water. Whether there's water or not, who cares? The pollen is going to get to the ovule. You get what I'm trying to say? That's a very nice question. Next thing. Why has the reducing... it's going to be a long day. Why has the gametophyte generation have been advancing to spermatophytes? So why is it that the gametophyte generation is an advantage, okay, is an advantage to spermatophytes? So in other words, why is it that it's better to have your pollen instead of having a prothallus? You know what a prothallus is? Can you remember? It's that heart shape, nice and fib, love, all that stuff. Heart shape, prothallus, okay? That thing, right, is if that thing dies, we're history, okay? If there's no water, it's very vulnerable. It can't get anywhere, okay? There's a big problem for that. So what we do is we put it into pollen, and that pollen can protect it, okay? It doesn't dry out. All it needs to do is dig that tunnel, remember. So we put a capsule around it, stop it from drying out, and it stays with the main part of the plant, the sporophyte generation, okay? It stays with the gametophyte generation. It stays with it. It all happens inside that. It doesn't have to be sent out and try and live by itself. Do you get what I'm trying to say? It's a very, 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 very important process, this. It's the best thing. The more complex the plant becomes, the more it needs protection. Think about it. In the old days, can you remember Nokia?
Yes.
What was it, the 911 or something, or the 1110, I don't know. You could throw that across the room.
3310.
That's it. You could bring it from this side, and you could chuck it around the room. It'll bounce off every wall, and you could still make a call, right? Nowadays, you drop your phone, and it's history. It's got more stuff in it. It's more complex, so it's very bad for it to drop. Do you get what I'm trying to say? It needs more protection. Okay, so that's exactly why it is like this, right? Let's have a look. Explain why most angiosperms, are able to produce less pollen than gymnosperms. I've just told you. Now, you don't know what an angiosperm is, right? I haven't taught it to you. We're going to learn about this one, this brother, right? This one next week, because it's so complicated and it's so cool that we're going to have an own lesson for it. I think that's brilliant, right? But angiosperms, these ones, right, are flowering plants. Why do they not have to produce as much sperm as gymnosperms? Can you remember we said that gymnosperms have air, right? Wind. They're sitting on the top of the tree, right? And there's another tree over there, and they don't chaff. They don't go, hey, bud, I'm going to send these things your way. Catch them. It's not going to do that. Okay? All he's going to do is the wind's going to blow, and he's going to sway in the wind like this, right? And the pollen's going to go, and pollen's going to fly through to the air and hopefully land on a female, right? Now, angiosperms says to a bee, come here, bee, stand here, I'll give you some pollen, right? He takes the pollen, the bee goes off, he lands on another plant of the same, and now the sperm is there. The pollen is already there. It's perfect. I mean, what more could you want, right? That is so cool. That is exactly the main reason. This is here, catch. This one is, you're going to get it.
Well, guys, we've come to the end of today's show. Thanks for joining me. Remember to visit our website, learnextra.co.za/live to get the notes and to watch the videos. And if you are stuck and you have any questions, send an email to helpdesk@learnextra.co.za and we will help you. Also, guys, we are dedicating the month of March to Library Month. Join us as we discuss some great books and some exciting competitions on the way. Goodbye.